Let us study another type of inheritance pattern, that is pleiotropy. And in pleiotropy, the gene is responsible for controlling more than one phenotypic character or phenotype. So, we are taking the example of synthesis of starch in pea. The pea plant has two allelic forms for this, that is B capital and small b. And it was found that a plant having the homozygous condition, or that means when both the allelic pair are same, the members of allelic pair are same, that is in the homozygous condition, the plants were capable of effective starch synthesis. They produce large sized grains and they produce round seeds. So if the homozygous condition with capital B was there, it was found that the plants were effective in starch production. Also, they produce round seeds and large sized grains. So these were the traits which were regulated in homozygous condition. However, if the small b pair, that is again the allelic pair, are same, and we know that this capital B is representing the dominant and the small is representing the recessive allelic form. So small b, again it is homozygous and it had less starch synthesis or less starch production as compared to that with homozygous capital B condition. Also, it produced small size grains and wrinkled seeds on maturation. So on maturation, it got round seeds, and over here, when homozygous recessive allelic form were present, then it produced wrinkled seeds. So these were the traits which were observed. However, when crossing took place and the heterozygous situation, it was found that it led to the production of intermediate sized grains and also round seeds on maturation. Now, what can we conclude from this? We find that this particular allele or gene is regulating and the size of the gene, size of the seed, and also the shape. So this dominant gene is actually dominant for shape of seeds, and it is because the B, always capital B, is producing the round seeds. So here it is dominant for the shape of the seeds, but in heterozygous condition it is unable to express itself or regulate the size of the grain. So here as a result the intermediate size grains are produced. So codominant, the B gene, which is a dominant gene, is showing dominancy for the shape of the seed, but it is codominance for the size of the grains. So we can conclude that here this gene B is controlling more than one phenotype character, where it is also regulating the shape of the seed and the size of the grains. But in heterozygous condition, it is unable to express itself completely for the size of the grain, because we are getting intermediate size grain, whereas if it is a homozygous condition, then we are getting large size grain. However, it shows complete dominance for round seeds, because the size of the seeds is again regulated by this. So it is this gene B is showing pleiotropy, where it is regulating more than one phenotype character, that is the size of the grain and also shape of the seed. So this is another type of inheritance pattern.